Okay, can you see my slides?
Yes, sir.
Okay, all right. So, is it my clear voice is clear to you guys?
Yes, sir.
Okay, all right. Um, Fine. So here we ended up with yesterday's class and trying to explain that what is hydrophobic effect. So yesterday what we have explained is what is bioorganic chemistry. That people who has अभिनव जी मेरा क्लास चल रहा है. Okay, so we started with what is bioorganic chemistry, and the most important thing, how the bioorganic chemistry, this concept has been generated. Further, I talked about the molecular recognition, which is the central event, I think, and many people think about, because if you look at the biological system, mostly the connectivity and any kind of physiological activity is going on through the specific interaction, which is known as molecular recognition. And I talked about define what is molecular recognition, also given the examples, one or two, and then talked about the non-covalent interaction. What are the non-covalent interaction? And here we ended up with the summarized table that the metallic bonds, which are in, you can say, like a intermediate bond strength of or bond energy, you can say, and covalent bonds. Ionic bonds are strengthiest, you can say, and healthier than compared to van der Waals or hydrogen bonds. But as I said, that the bundles of sticks are very difficult to break compared to a single stick. So when you have a cumulative or a bunch of hydrogen bonds or van der Waals interaction that allows strain their bond, and let me see if someone would like to join and then not join it. Okay, let me allow. Fine. Okay, now the last point that talked about the hydrophobic effect. So hydrophobic effect is desire for non-polar molecules. So it's not polar at all, as you can understand. It's a greasy-like. Okay, so when a greasy-like molecule tends to be, be in the water in aqueous solution, so they try to aggregate in order to separate from the water. So that particular phenomenon is known as a hydrophobic effect. So the best example that I have given, that how a gecko walks in the water. So they have a very individual microspatula, and this microspatula, you can see one of the kind of microscopic structure, how it looks like. They just basically keep walking on the wall by using this hydrophobic interaction. This is van der Waals forces, as you can say, grabbing the structure I'm showing here, as you can see. It's a kind of a microscopic cartoon. Okay. So next, that what do you wanted to know about the molecular recognition? That solvent effects. This is one of the very important parameters that solvent play a dominant indirect role in driving molecular recognition in solution. What does it mean? That solvent, maybe if it is a, let's say you are thinking or trying to design a molecule that binds to any other molecule. Let me draw in the board that you can understand. So let's say this is one molecule would like to bind. You can think about that is another molecule. It binds here. Now, if this particular binding works in, let's say, chloroform,
Sir, could you please switch to
it is going to
blackboard?
bind. Hello?
Sir, sir, could you please switch to blackboard? You are showing current presentation, the slide.
Just 1 minute. Huh? Sorry, just let me. Can you see the blackboard?
No, sir.
You can't see blackboard?
No, sir.
Anyways, this is not blackboard. Anyways, this is whiteboard.
Yeah. Now we can
Into
see.
the confirm
All right, let me share it. Okay. Now it is partial. Okay. You will be able to see both slide as well as the board. Can you see?
No, sir. Currently, your slide is open.
Huh? Just slide. You can see only slide.
Yes, sir.
Oh, that is interesting. I don't know. Well,
We can see, sir.
my whiteboard.
Yes,
Sir,
sir.
it is visible. Actually,
Huh?
Yes, sir. Your whiteboard was visible.
So he has to pin up in the screen, sir. He has to pin up on the screen. There are 2 slides.
Let me see. So what am I saying? I need to. You need pin up screen. Pin up screen.
there's an option on the screen to pin up. Then the main screen will appear. Your video will be appearing on the main screen.
Yeah, tell me again. I can't hear properly.
So there is a pin up option on your screen.
Pin up option in the screen.
Yes, sir.
In the middle of the screen, move your cursor, then it will appear.
Yes, sir.
स्टूडेंट्स को चेंज करना पड़ेगा सर के चेंज करने से नहीं होगा
yes, sir. It's one's individual choice. Means, Nitesh Mani, bhaiya, you can pin up on your screen. That
Students has to pin up. Someone is saying.
Yes,
yes, sir.
yes sir actually when we pin up the screen then your board will be visible and when your presentation is pinned up then the presentation will be visible.
Okay, I see. Okay. So what you see now is it just my right?
Whiteboard only.
Whiteboard only.
Whiteboard.
Whiteboard only.
Yes, sir.
Okay.
So you don't need to do anything, sir. Student will do.
Okay.
Yes, sir. Because there are 2 options from your name. One is having the whiteboard and other is having the presentation. We have to unpin the program. Like if we want to see the blackboard, we have to remove the presentation and then pin the whiteboard. We have to, make
So
the alternate.
right now I am sharing the only slide. Okay. Is it fine now? Slide, can you see?
yes, sir. Yes, sir.
Okay. Okay, let me talk in slide. I don't know. It's I have to figure it out how to switch these things. You guys, I'm just sharing both actually, half and half window. I'm trying. I have shared half and half window, but I don't know why you guys can see that. Um, Let me just stop this and then present now. My recording is already started. If I do my entire screen, then only one screen will show. But if I say a window, then it should show both. Okay, all right. Can you see now my okay? So you can see now the presentation, right?
Yes, sir.
Okay, and you can see my presentation as well as you can see my whiteboard also. Isn't it? Or not?
Yes, sir.
Good.
Sir,
Now
the
you can see.
presentation is not completely shown.
Yeah, that I will manage it. That I will manage it. Now you can see completely, right?
Yeah.
Yes, sir.
Yeah. So that I will manage it. So just I wanted to just first make the screen adjust. Okay. All right. So now let's take a look at what I was talking about. So here is the thing that let's say this is a guest molecule, and this is the host molecule, and this is the guest. Let's say this is the host molecule has to grab the guest molecule in chloroform. So you have tested this hypothesis, it's working, but when you add the water, it's not, this particular recognition is not working. That's what I mean here. The solvent can play a huge role. The reason why it is because the non-covalent interaction, as I already talked about the hydrogen bonds, ionic interaction, all these things can be hijacked by the water. As you can see, the water molecule, look at this water molecule has capability of forming 4 hydrogen bonds with another 4 molecules. That is the way the water molecule make clusters. It has an enormous power and rich source of hydrogen donor and acceptor property that you already know about, how water works, how water solvents, any ions, any other molecules. So that is the reason, and it goes through mostly like a hydrogen bonding. And so what is going to happen, let's say a guest has to come and bind to host. It may not be possible in water. The reason behind, let's say if guest has to interact with the host through a hydrogen bonding or some ionic interaction, that already it's a huge amount of water. It's a soluble, some molecule soluble in water. That means a concentration of particular that molecule will be very lower compared to the concentration of water. What is the concentration of water? Tell me, anyone.
55.5.
55.5, right? See the concentration, that's molar. So that concentration is very high. So it will occupy most of the place of this gas. So that interaction will be hijacked. That is the reason that it's not necessarily that it's going to work in water. So it plays a very like role. It depends on solvent to solvent. But so now you look at that the importance of molecular recognition in biological system, which is the most important thing that we wanted to talk about. I have given the basic principles, talked about. Platform has been already established. What is molecular recognition? Now you come to the biological system. I'm not going to go very details what is happening, many biomolecules. Just wanted to give the idea what is happening. So molecular recognition plays an important role. I would say like a central events in biological system observed in between the receptor ligand binding, antigen antibody, DNA protein, protein, sugar, another protein is known as a very specific for the sugar, known as a lectin, RNA, ribosome, etc., etc. As the given example, you can see the virus, how it goes inside. Again, it's a protein to protein interaction. It interacts with the protein and it has a specific receptor. It binds and further goes through the many physiological, conformational change and many other kind of changes. And that is the way virus deliver its material to the in our act cell, which hijack our cellular system to make a more virus. You guys already know about virus, and it's now the thing has come that everybody knows about that. Virus is not a living system, you know that. Okay, it's just a assembled particle having biological molecules, and such a fancy way it has been assembled that if it once it goes to our cellular system, it hijacks our machinery to make many virus particles. And it's a very fantastic and very complicated mechanism. But ultimately, you see the major event, what you see, which is molecular recognition. If some people do not have the proper, that receptor, that virus can go and just do this kind of activity, that people may not be, infected. But generally, if virus, it recognize such a molecule that most of the, almost like a 99.999, you can say, people already have this kind of receptor. Like, as you can see, the bacteria also does similar kind of thing. And now question becomes, some people are asymptotic. Asymptotes there. Okay, so that means even the virus infected, they do not have any kind of symptoms or any problem, any kind of things. Why is it so? Because these people have some particular antibody. It's another type of molecule, which is our immune system, produced by the plasma cells, mainly that red blood cells in our body. Okay, and what happens? This antibody can recognize the virus. Again, it's a molecular recognition. It's a very specific recognition that neutralizes the viruses. Why I'm saying the neutralizing? And that is the exact word in biology. The neutralization. Neutralization means it actually it is lethal. This particular virus, you know that, and it neutralizes. Antibody neutralizes that. So now you look at one by one some example. This is the example given yesterday, that molecular recognition can be established through the last resort antibiotics, natural product, vancomycin. This is the crystal structure, what I am showing here. As you can see, the blue color is the vancomycin. It's a cyclic peptide. What is that full screen?
So the presentation, sir.
If I make a full screen, can you be able to see my board then? That's the question.
Yes, sir.
Okay. Can you see my board now?
Yes, sir.
Okay, that's good then. That's good. Okay, all right. So, so now let's look at that. Yesterday I was talking about what is peptidoglycan. So, the structure you look at, yesterday I was drawing. So, this is the main part, which is glycan. The bigger, balls are. The glycan, these are glycan, and small peptide, these are the amino acid, like lysine, glutamate, lysine, D-alanine, and another D-alanine. So D-alanine at the end is the component going to recognized by vancomycin. So what happens? That particular the terminal D-alanine, D-alanyl D-alanine, that means the two D-alanine threaded with the two amino acids, a small peptide, in a bacterial cells through 5 hydrogen bond it binds. That yesterday we talked about. As you can see in the structure, the vancomycin is lethal to bacteria since once it has bound to these particular peptides, they are unable to use to construct the bacterial cell wall. That yesterday I already discussed the same thing. See how important it is. Whatever you see, the drugs, they are working. Most of the drugs, it binds to a particular receptor. Again, it is a specific recognition. Some drugs, they do not do what you think. That okay, it binds to the receptor. No, not that is the way being recovered, or we just be cured by that. Not all drugs work like this way. Like as an example, I can tell you that you take paracetamol, but paracetamol it doesn't have proper like a receptor kind of thing. Okay, so but as an example, I can give another drug. It can just the remdesivir recently. You are sure about that it works against some particular viruses because it blocks a particular enzyme so that the virus cannot replicate. Okay. Now, look at, here is the thing, why I was talking about that. The solvent is important. The general features of protein-ligand interaction. Let's say protein and ligand, how it interacts. So, let's say the green one is a ligand and the cyan one is a protein. They form a complex. This complex is completely reversible, though this is not shown as a reversible, but it is a reversible. I am showing here only the association, not the dissociation, but it is a reversible complex. So I'm showing the association constant, which is Ka. Okay, so that tells you the equilibrium constant actually. What is that? Kinetics of your reaction of the association as well as the dissociation. Okay, so that is the way you can define the Ka, association constant. And also you can define as a dissociation constant Kd, the backward direction. Same thing, just if any particular system is in equilibrium, specifically what the thing I'm showing, the two particular molecule, they are forming a complex, you can say that this is Ka equal to 1 by Kb, or you can similarly say Kb equal to 1 by Kb. Right? I hope this is you can look at any searching the either Wikipedia or any kind of literature. So optimizing protein binding affinity by a ligand is the crucial steps in the drug discovery. So protein binding affinity means what is the strength of this binding? How strongly it is binding, that tells about the drug's efficacy. It's not only that is the only parameter, but it is one of the crucial parameters. The reason behind that how much material you need to, let's say, add in a particular cellular system or in a in vivo system that will work. Let's say the affinity is very low, then you have to take a really that much amount of material, which is going to cause a problem in your body. But affinity, if it is very high, a small amount, or if it is very specific, the small amount material can work. That is the thing. So affinity is one of the things, a crucial step in the drug discovery. And this is one of the parameters for molecular recognition that we talked about. Let's say how it works. A ligand binds to protein complex, how it works in the water system. Protein, if it is completely solvated, you know that. So any molecule it goes to water, it's completely solvated. That means if it is like consider this is a protein, there are many functional groups in the surface that interacts with the water, having a hydrogen bond, and it's solvated protein, you can say. So now you look at this particular cavity, it's already occupied by the water. Now you have to make such a ligand that can drive some hydrophobic interaction in the pocket so that they can go and sit and replace the water, and finally they can make a solvated complex. So in that way, you have to replace some of water molecule, as you can see here. So to replace this water molecule, you have to have a property of ligand so that it can occupy and force the water molecule out. That means there should be a proper driving force or proper van der Waals or non-covalent interaction forces, the accumulative, or you can say a cumulative way, they can replace the water molecule from the surface of the protein pocket so that it can bind. But of course, it will bind specifically whenever it's going to obey the particular parameters I talked about. Like it has to have a complementary. One of the important parameter it has to be a complementary. Now look at that pre-organizing of a ligand structure is one of the major criteria. This is, let's say, if you want to design a particular molecular recognition technique, I'll show you what are the applications. This is the most important thing in the world is happening, I can tell you. You will realize the thing what is happening. Either you are going through the diagnosis process, you are designing drugs, you are designing any kind of sensors, you are going through many other things. This is molecular recognition is one of the big parameters. That's why 1987 Professor Ling got a Nobel Prize. All right, look at, this is the protein, let's say. Now you have a flexible ligand. This flexible ligand is going to bind with this protein once it will reorganize through the conformational changes. Like this. Then only it can bind. But it has a conformational freedom, lot of freedom. Okay, now in that case, you are getting some particular DLG value of this complex formation or association constant. Now, you know that this particular ligand needs to have these two in a same direction, in a particular direction. How could you reorganize them? If you make a bond between this particular two center, it's going to form this kind of molecule. So now it is pre-organized. No need to go through these conformational changes. It will immediately come and bind because it's already set. Now question becomes here, the pre-organism of ligand structure is one of the major criteria. Why I'm saying so? Now see, this here you need to pre-organize by the conformational freedom, by the ligand itself. But here you did the pre-organization and it is binding. Now I am asking the question that which one is going to be the most favorable process in terms of going to be a better complex formation. So this one is going to be the better complex formation than this one. Why is it so? Because as you look at here, the del S, the equation you know very well, this equation. The del S value, this was a very random and it pre-organized to this particular thing. The del S value will be higher. Then it's already pre-organized and the same pre-organized it binds. So the randomness is not going to change much. This entropy here, it is going to contribute a less amount than this one. So if you think about the del H is some particular value of a negative charge, the negative value, let me find the this particular equation, del G equal to del H minus T del S. Now you look at, this is constant, let's say constant is, I'm giving you some value like a 600 something. And now this one is T. Now this is going to tell you what it is. So del S is going to be something like a minus, now bigger, now X I'm writing, okay? So now here in this case, when it is not pre-organized, case of like the first one, let's say it is A, and the second one which is pre-organized is A dot. Okay, so in this case what is going to happen? So A is greater than the A prime, this particular value. So when it is more, it's going to be more positive value. That's going to decrease the value of the total del G. So total del G value decreased means the stability of the complex. Ultimately, eventually you can define as the equilibrium constant of association is going to be lower value. So that means when you have a pre-organized structure that can allow you to generate a better stability complex. So that's why the pre-organization of the ligand structure is one of the major criteria when you design any particular host or guest molecule for a particular purposes, either drug design or any kind of molecule host guest supramolecular chemistry you would like to do it. That's the particular thing. Sensor you would like to design. So these things we will discuss more about in a cyclic peptide section. That when we talk about peptide and cyclic peptide, nature produces cyclic peptide. The reason behind like this, entropy cost of binding. Okay. So the molecular recognition so far people have seen in the biological system is biotin and streptavidin is one of the, like I would say, is example that people generally always raise. This example is the best binding you can see between the ligand and protein. Its affinity is femtomolar. If you leave a femtomolar concentration of biotin to the streptavidin, it's going to bind. Interaction is very, very strong. And that's why people consider this as a kind of, it's equivalent to a kind of covalent bond. And this recognition process, people have been utilizing for many purposes in research. These complexes widely used in the non-covalent conjugation in biological, biophysical experiment, like if you want to, let's say, conjugate any fluorophore to the protein. How do you do that? That protein, let me tell you here itself. Let's say you have a protein that you want to do the biophysical studies of this molecule, and how it behaves, how it's the dynamic, other things you want to do. So how do? You need to have at some point, having a sensing parameter. So fluorophores, you know about it, what is fluorophore? And these fluorophores people tag it. So now how they tag it? They tag it with the streptavidin, they put it strip, and then biotin, and then put a fluoro. That is the one way they can conjugate, and they can study that what is happening with it. And many other process actually, and biotin for the purification purposes also. Let's say you want to purify this molecule, it tags with the biotin, and then you have a streptavidin column pass through it, so there will be interaction between the streptavidin and biotin. This protein will be stick into the column. Other things you can elute it, and then further what you do, denature this streptavidin protein, so that is the way you can elute it. That's a different kind of strategy people use it. Okay. Another biological recognition, most important thing is transcription factor. The gene expression. Why do we need that gene expression? Just because of your physiological process. Let's say you need to have a wound healing. Somewhere you have a cut. So it has to, somewhat go the signal. DNA has to finally do the gene expression as a function of a production protein. That protein can recover that particular place by the relevance molecule. So it needs the interaction between the protein and DNA. How it happens? Because DNA is sitting. There are some active protein, it goes and bind to the DNA. That triggers particular signal. That signal allows to go for the enzymatic process, allows to make it a gene expression. The DNA to mRNA, mRNA to further it goes through the translation process, make a protein. That's the way it happens. So DNA, basically, if you look at the structure here, DNA is basically bound with a histone protein. And these are not like a specific just interaction by some point of just ionic interaction. But here, what happens in this particular pocket, some protein comes and interact and give a signal that, hey, something you need to do it, production of that particular protein. Do it. That's what it gives a signal. That's call it gene expression process. And this gene expression process require the specific interaction between the protein and DNA. If something goes wrong, cell is going to produce something different. It can finally give you either a tumor, cancer, or something kind of other diseases. So it has to have a specific, the network has to be very, very strong. And every day, if you look at a simple cell, you are a big, big organism, but a cell simply does such a millions of this interaction to run the process. Protein-protein interaction is the another thing that is happening everywhere in the body. And whatever your things happening in like life is most of the physiological process responsible for the protein-protein interactions. And these people have realized lately that protein-protein interaction can be, this understanding of protein-protein interaction can be used for the drug design. So some protein-protein interactions goes out of control. So you need, something which is not in a proper limit. is going to be always a problem. So protein-protein interaction also in our body has a proper network, proper connection, proper interaction, and it has a particular limitation, a particular threshold. And beyond that, when it goes, it's going to have some problem. So one of the example of protein-protein interaction is just I'm saying the signaling molecules. Let's say a cell, outside the cell, some protein comes and signal it to do something, some other job to that cell. This kind of things, protein-protein interaction plays a big role. Now, as an example, I'm going to give you the how protein-protein interaction can cause the cancer. Let's look at this. This is very well established thing which is known as MDM2-P53 interaction. Nothing to read about. MDM2, it's one kind of protein. P53 is one kind of protein. They interact each other. And why it is P53 named? Now it's a P53 kilodalton is the protein's molecular weight. That's it. People have seen in biological study that this is one of the protein known as the guardian of genome. Guardian means it controls, it advocates everything. Hey, you want to do it this. If your cell needs to die, you have to die. This cell has some problem in the DNA, otherwise it's going to cause some cancer or some other kind of problem. Hey, cell, you should die. Everything. It's a guardian means it does through the genome. So it is mainly known as a tumor suppression. So that means it is clear to you. So this particular protein advocate gene such a way that if by any chance any problem comes into the DNA, so that can cause a tumor or cancer, it will program such a way so that cell can die. Beautifully it has been controlled and it's very complex. But what happens, if P53 continuously say to DNA cell die, so then how cell is going to survive? So someone has to control that. Guardian has another guardian, of course. So that particular MDM2 is another protein. What happens? It just interacts with a specific N-terminal interaction of P53 that allows the specific interaction, again through a molecular recognition, allows to control the P53 when it needs to release, and if it's not required, let it be inactive. So the cycle I'm showing here, let's say there is a DNA damage or cell cycle abnormalities. Cell cycle abnormalities means you can understand that one cell cycle, cell divide, it can happen that there is a problem in terms of the DNA copy. When they make a DNA copy, there's a problem somewhere, there is a change or mutation. So that's going to lead to some other disease. There are some checkpoints. So that particular checkpoints, this P53 controls everything. That checkpoints, the P53 realize that if there is some problem, automatically what happens, the P53 releases through some enzymatic pathway or some different kind of protein triggers that, okay, P53, you are inactive, you have to be active now to go to interact with the DNA as the interaction I'm showing here, and then it just need to you do what it is. Either you need to do the DNA repair or cell cycle arrest or do the apoptosis. Apoptosis means cell need to go for the programmed cell death. Cell need to go for a programmed cell death. It's not just cell death, it's a programmed. That means that it goes through the programming process. Again, the program is here interaction. This interact to this interact to other protein and says that, okay, you need to die somehow. They have a mechanism. Look at the molecular recognition and supramolecular chemistry that has been developed in the lab. There's a very interesting thing that you guys know already about, that how specific crown size, crown ethers, they recognizes different atoms like lithium, sodium, potassium, rubidium, cesium. As you can see, in a periodical table that they're increasing their cationic radius, you need to increase the shape of the molecule. That is already we discussed. That shape is very important. And also, along with a proper orientation and proper connection. That means either ionic interaction or hydrogen bonding. That is very important. So as you can see, it's very nicely constructed such a way that lithium can interact with the oxygen. Okay, it can have a ionic interaction between the oxygen lone pair and the lithium. You can see the 15 crown 5, why the name like that? 12 crown 4, 4 means the 4 oxygen, 12 means there are total 12 atoms. 15 crown 5 is the same thing, 18 crown 6. So now the question becomes that what are the things to learn here? So one thing you can learn that the shape is very important, and also another thing is most important thing, that a proper choice of the functional group. Look at it would have something else. The oxygen could be the best choice. That's why it has been taken. And why it is oxygen? That because this ether link is going to make a certain kind of the receptor shape or particular orientation. See, many things matters. So altogether, it's a tuning of the property such a way that it can behave as a functional group, as it's a shape, as well as its proper orientation. Okay, and also it fits into the topic. So it's a complementary. That's the thing. It has to be properly designed. Then only you can see that. One example that I'm giving. You guys know about the antibody very well. Its antibody is a Y shape. As you can see, it's a Y shape biomolecule. And the feature of the antibody, you already know about it. It neutralizes pathogens in our body. It's our mainly our major defense system in our through the immune system. So as you can see, I can describe that this part is known as the FC region. Okay, this whole this big body, nothing it does, just this portion. It looks like a loop size, like a loop. This you can see. Look at the loops. How loops looks like in that particular. This portion is the mainly responsible for binding with the antigen. Antigen means there would be virus, it could be different kind of pathogens, bacteria, many other foreign molecules, many things. So this is the mainly responsible for the binding. Now, people also tried to mimic that also. It's an antibody mimic. How could you do that? People made a molecule, it's known as a peptide. It's a little bit different from the peptide. We'll discuss during the peptide description. Now, such a way it has been described, synthesized or designed that in the air-water interface, they make a kind of a loop, as you can see. So once the molecule they synthesize, they drop into the water, and this water, as if these are, you can see the phenylalanine, they not phenylalanine, sorry, the phenyl groups, I would say. Phenyl groups, these phenyl groups, they want to avoid from the water. But in the opposite side, you can see the amine and carboxylic acid. They are the polar group. They would like to dip into the water. So that such a way it has been designed that air-water interface, there in between, there are some nice functional groups. They make a loop kind of thing. And totally they take a shape of like this. And people have designed such a nice kind of functional group in the air-water interface that it can inhibit the protease particular enzyme activity. Again, enzyme works for a particular reason. They do catalysis in our body, do the reaction, but sometimes it goes out of control. As I said, it has a particular threshold or particular a limit of their activity. If they cross their limit, automatically it causes the diseases. So that time you need to inactivate that enzymes. That's why we use drug to inactivate them. Now, in this case, that people have used some particular this kind of design to inactivate. So just I'm showing the antibody mimic how people can do it for the molecular recognition purposes. Molecular recognition are two types. One generally static, second is dynamic. Static is very simple. Just an example, as you can see, that you have a host, guest come and sit, exactly fit. Dynamic is little bit different. Dynamic is the molecule is not, capable of binding that particular molecule, like the green. Let's say any guest you would like to bind, you would like to make it to bound here. It is not going to possible until unless this red color, another molecule, binds to that pocket. So what happens? First it binds and rearrange. Rearrange that particular, the host shape, again, complementary shape. Remember that. It's a complementary shape so that it can fit very well and recognize tightly. That's a dynamic. Static is just like a one-to-one, and dynamic is, it needs a cooperativity binding to the other molecule. All right. Now, if I ask you the example, what is that? Example of this, you can easily say the crown ether. Let's say you can say 12 crown 4 is going to specifically recognize the lithium ion. This is exactly the static. Dynamic is positive allosteric activation. What is this? Positive allosteric activation. Some people, you know about the enzyme. If you know about the enzyme, you know what is allosteric. Okay, so allosteric is the one of the kind of pocket in the enzyme other than the active site. Okay. So that particular pocket or particular, let's say, surface, if any molecule binds, then only this enzyme gets activated. Otherwise, till that, enzyme is not active. Okay, so see that this is the one of the best example. That means, let's say any enzyme needs to activate and do certain work. If it is a positive allosteric activation, it goes through. Some molecule has to come and then bind, then only it will be activated and do the work. So it's such a nice programmed and very complicated network, or you can say very sophisticated, delicate network. Our nature has been produced through the evolutionary process. Not happened in one day. It took billions of years to do that. Okay, look at the molecular recognition applications. You can design sensors for, let's say, cation, any kind of molecule you would like to sense, any, let's say, pesticides you would like to do, any cations, okay? Enzyme mimetic catalysis, you can do it. Self-organization, how do you do that? All these things matters, okay? Just a couple of examples that I'm showing here. Showing some example of the crown ether. Let's say it attached to some particular protein. Now, once it is, let's say, bound to the calcium, this is one of the example. It gives a lambda max is 490. However, when it is barium, it's going to be 560. Okay, so these are the kind of examples that if you attach to some fluorophores, they can be activated or change their wavelength of maximum absorption. This one has an absorption at 477. Once it binds to calcium, it goes to the 490. And once it binds to the barium, it goes to the 560. Similarly, as you can see, these are the another, it can accept this kind of ammonium cation, the ammonium cation group. So that can allows to make it some turn on fluorescence detection. All right. So Now the thing is, I would say that I'll stop here. The molecular recognition. This is one of the kind of elective course. Generally, the next semester floats by our department. If someone is interested, I would say that you can go and take it. You will learn more and more things because the application-wise, if you see the molecular recognition, if you understand properly, you can design many things in any direction. Either you could be a very good organic chemist that design an organic catalysis, either you can design a sensors, either you could be very good drug designer, either you could be a very good diagnostic designer, either you could be very good a very supramolecular chemist. This is a very interesting topic, molecular recognition, which has been just recently being a part of in the academic curricular activity. It's not because it has been discovered just like 50 years ago. Okay, and it took some time to come into the as a book or academic curriculum activity. So therefore, I would say that this is one of the very important thing you can focus or look into it. So we will slowly discuss more things.